Welcome to this series dedicated to cell biology. Today, we will dive into the topic of actin microfilaments. If you're new here, consider subscribing and checking out our Instagram and Facebook pages. Actin microfilaments, shown here in green, are one of the three main types of cytoskeletal filaments, alongside intermediate filaments and microtubules. These microfilaments are composed of F-actin, a structure formed by the assembly of G-actin monomers. In other words, F-actin results from the polymerization of G-actin. G-actin, or globular actin, is the monomeric form of actin, while F-actin, or filamentous actin, is the polymerized form of G-actin, forming long helical filaments. Actin is one of the most abundant cellular proteins. It constitutes 1 to 5% of the total protein content in non-muscle cells and 20% in muscle cells. The actin filament exhibits polarity, with a plus end and a minus end. This figure shows the stages of actin microfilament polymerization, where G-actin monomers bind together to form an F-actin filament. Each actin monomer is bound to an ATP molecule, in red, which is hydrolyzed to ADP, in blue, after the monomer is incorporated into the filament. The ends of the filament are marked plus and minus. The plus end is more dynamic and promotes the addition of ATP G-actin monomers. The minus end is more stable and primarily sees the dissociation of ADP G-actin monomers. ADP G-actin monomers mainly add to the plus end of the filament, as shown in the first part of the image. After addition to the filament, ATP is hydrolyzed to ADP, blue monomers, which weakens the binding of the monomers to the filament. Then, over time, ADP G-actin monomers dissociate from the minus end. In summary, this figure illustrates the dynamic cycle of actin assembly and disassembly, where the energy provided by ATP is used to maintain a balance between polymerization at one end and depolymerization at the other. This process of polymerization at the plus end and depolymerization at the minus end is a mechanism by which actin filaments remain in constant reorganization while maintaining a relatively stable overall length. This mechanism can be disrupted by exogenous substances such as certain drugs that affect the polymerization and depolymerization of actin microfilaments. Cytochalassins, derived from molds, inhibit AMF polymerization by binding to their positive end. Phalloidins, toxins produced by fungi of the genus Amanita, block AMF depolymerization by binding along their sides. Depending on their function, microfilaments are organized differently, either into bundles or into a network with gel-like properties. They interact with specific proteins called actin-binding proteins, ABPs, each possessing unique properties, in polymerization and depolymerization, stabilization, bundle and network formation, membrane attachment, fragmentation, etc. To illustrate the diversity of actin microfilament organizations in the cell, let's examine a specific example of actin arrangement in epithelial cells. As shown in the image, microfilaments are grouped into parallel bundles in the apical region of the cells, forming structures known as microvilli. Each microvillus contains parallel actin microfilaments, anchored to the plasma membrane through an anchoring protein complex, myosin-icelmodulin. The actin filaments of the microvillus are connected to each other by protein linkers composed of vilin, fimbrin, or facin. They insert into a terminal web, a specialization of the actin cytoskeleton, located just beneath the apical plasma membrane of the epithelial cell, and connected to the plasma membrane via the spectrin protein. These microfilaments provide stability to the microvilli. To understand the importance of this actin microfilament organization, it is essential to examine the various functions it enables the cell to perform. Actin microfilaments play a key role in the movement of organelles within the cell. This movement is facilitated by the association of microfilaments with type I myosin. This protein binds organelles, such as vesicles, with its tail and uses ATP energy to move along actin microfilaments with its motor head. In this way, organelles can be efficiently transported within the cytoplasm of the cell, contributing to the distribution of essential cellular components. Another key interaction between actin and myosin involves type II myosin. Unlike myosin I, myosin II acts on actin filaments oriented in opposite directions, causing the filaments to slide toward each other. This movement occurs through a contraction mechanism, where the release of ADP initiates a shift toward the plus end of the filament, resulting in a shortening of the distance between actin filaments. These dynamic structures are not limited to intracellular movements, but are involved in a wide range of cellular processes. Let's examine these functions in detail to better understand the importance of actin microfilaments in cellular life. Let's start with one of the most emblematic functions of actin microfilaments, muscle contraction. Muscle cells are characterized by a contractile apparatus, consisting of the end-to-end association of contractile units called sarcomeres. In electron microscopy, these sarcomeres give the cell a striated appearance, with alternating light and dark bands. The striated appearance of the sarcomeres reflects the precise organization of actin and myosin filaments within each contractile unit. It is precisely this regular arrangement that enables the coordinated interaction between actin filaments and type II myosin, a fundamental mechanism for muscle contraction. The roles of these actin microfilaments are not limited to muscle cells. In fact, they also play a role in other contractile processes within non-muscle cells. Two notable examples are stress fibers in fibroblasts and the contractile ring during cytokinesis. These mechanisms, although distinct from muscle contraction, rely on the same fundamental principles of interaction between actin and other proteins, such as myosin, to regulate cell shape and dynamics. Another essential role is manifested in epithelial cells, through the formation of the adherens junction. Indeed, actin microfilaments assemble beneath the plasma membrane at the apical level to form this belt, allowing contractile forces to be transmitted across cells. This coordinated mechanism plays a key role in maintaining the integrity and structure of epithelial tissues. The fifth role of actin microfilaments involves cell migration. The image illustrates the movement of a cell, highlighting actin polymerization at the lamellipodia at the cell's leading edge, where the actin cortex is under tension. This process allows the cell to extend forward, while the retraction of the cell's rear, coupled with the formation of focal adhesions, enables coordinated movement. This mechanism is fundamental for processes such as wound healing and immune cell migration. The final role of actin microfilaments is in phagocytosis. When a phagocytic cell encounters a bacterium, a signal is triggered involving calcium and gelzolin. This signal leads to the temporary disassembly of actin filaments, transition from F-actin to G-actin, from a gel state to a sol state, allowing the membrane to remodel and engulf the bacterium. Subsequently, actin filament polymerization reestablishes a dense network that forms filopodia, membrane extensions that enable the cell to capture and internalize the bacterium for degradation.